Comparative Health Risk Assessment of JUUL System and Combustible Cigarettes. Combusted cigarettes deliver nicotine via smoke, which is formed from burning of the tobacco filler materials, paper, and non-tobacco ingredients. This results in a highly complex mixture containing over 5,000 chemicals, and to date, the US FDA has identified 93 of those compounds as harmful or potentially harmful constituents, or HPHCs. By comparison, the JUUL system is a pre-filled, cartridge-based electronic nicotine delivery system, which aerosolizes the nicotine-containing e-liquid using a temperature-controlled device to minimize thermal degradation byproducts of the e-liquid ingredients and the formation of HPHCs. As part of our broad research program, several studies have been conducted to determine the potential reduction in individual health hazard and subsequent reduction in population-level harm associated with switching to the JUUL system. The objective of this study was to evaluate the reduction in aerosol levels of HPHCs and associated non-cancer hazards and cancer risk from use of the JUUL 5% Virginia tobacco and 5% menthol products relative to smoke from combusted cigarettes. The levels of HPHCs were analyzed in aerosols generated from the JUUL system using both non-intense and intense puffing regimens. These were compared to published levels of HPHCs in smoke generated from the 3R4F reference cigarettes using both ISO and ISO intense smoking regimens. The majority of the HPHCs evaluated in the JUUL system aerosols were either below the levels of detection, BLOD, or below the level of quantitation, BLOQ. With the exception of water and base e-liquid ingredients, glycidol, formaldehyde, and acetaldehyde were the only constituents measured above the LOQ in aerosol from the JUUL 5% Virginia tobacco products. The reduction in aerosol levels of these HPHCs was greater than 94% as compared to the levels of these constituents in smoke from the 3R4F comparator cigarette. Similarly, substantial reductions in HPHCs relative to cigarette smoke were also observed in aerosols from the 5% menthol products. As shown in these graphs, glycidol in aerosols generated using the intense puffing regimen was 95% lower relative to smoke from the 3R4F cigarettes. The reduction in the other measured HPHCs was greater than 95% in aerosol compared to smoke from the reference cigarette in both the intense and non-intense puffing regimens. The potential reduction in cancer risk and non-cancer hazards from use of the JUUL system compared to cigarette smoking can be evaluated using an analysis that is focused on the HPHC yields, their respective toxicity, and magnitude of decrease between the levels in aerosols generated from the JUUL system compared to cigarette smoke. Of the measured HPHCs, 9, specifically acrolein, carbon monoxide, 1,3-butadiene, acetaldehyde, benzene, formaldehyde, benzoylpyrene, NNN, and NNK, were identified by the WHO to be the greatest contributors to the non-cancer hazard and cancer risk from cigarette smoke and were recommended for mandated lowering. In the JUUL 5% Virginia tobacco aerosols, 7 of these constituents were either below the level of detection or quantitation and would not be measured. Although formaldehyde and acetaldehyde were present in aerosols above the LOQ, the analytical chemistry data show greater than 94% reduction in all of these toxicants compared to cigarette smoke. The analytical chemistry from aerosols generated from the JUUL 5% menthol products also shows substantial reductions in the 9 priority toxicants identified by the WHO. Although aerosol formaldehyde and acetaldehyde levels were above the LOQ, the levels of these toxicants in the JUUL system aerosol show greater than 98% reductions relative to cigarette smoke. The lower levels of these potent HPHCs in the JUUL system aerosols compared to cigarette smoke demonstrate likely substantial reductions in the potential exposure and associated health risks from use of the JUUL system compared to combusted cigarettes. In conclusion, the aerosol produced from the JUUL system showed a marked reduction in the overall number and levels of HPHCs. The decrease in the number and levels of HPHCs in the JUUL system aerosols demonstrate likely substantial reduction in toxicant exposure and the overall associated health hazards, cancer risk, and non-cancer hazard as compared to cigarette smoking.